So now that we have seen that how the marine microbiological work has been initiated and where we are today, right? So let us quickly start looking at how do we study them. Okay. The whole methods in microbiology can be divided in 5 different parts. The first one, which is almost common in all the methods you go further, right? That is the sampling method. The second one is trying to cultivate the microbes. Third one, utilizing the imaging techniques, that includes microscopy, that includes any of the fluorescent technologies. Molecular based method, again, utilizing gene sequences, or let's say DNA and RNA based studies. And then we have radioisotopes for the ecological interactions. So what it was originated in 50s. Okay, we will go one by one, and let us go in the sampling methods. So the sampling method for marine microbiology is the very first step, that how you start the understanding marine microbes, right? The most easiest way is to put the plankton net in the water and do the filtration. So you can see that one researcher is ready in the boat with the net. Okay, this net looks something like this, where you have the nets and then you have a bottle. Okay, so the water will go in from here. Anything smaller than that particular net size, right, will go out, and bigger than that net will start accumulating in the bottle. If you remember, what I taught is that plankton can be of different size. It can be femtoplankton, it can be nanoplankton, microplankton, mesoplankton, and so on, right? The size of this maze depend on what you want to study. If you want to study a plankton with a lesser than 35 micron in size, you will use this mesh size, which is available in different mesh sizes. Anything bigger, I mean, if you want to filter that 35 micron plankton, you will use this mesh accordingly, right? Just for your understanding, I have put here a picture which says that there are different mesh sizes available for this plankton. Okay, and this plankton can be used mostly to collect phytoplankton and the zooplankton. Okay, all those bacteria which is associated with the phytoplankton or the zooplankton will also be, since they are attached to the phyto and zooplankton, will be collected in this bottle. Okay, and this is how you tow the net. Okay, I hope that's clear. So the first sampling strategy is utilizing planktons nets, and the plankton net mesh size will depend on the type of organism you are interested in. Okay, that is for the water column. But what if you want to study the bacteria of which are present in the sediments? So for that purpose, you have grabs and the corers. So the grab looks something like this, okay, which will grow. If it is a sediment, you put your grab here, it will collect the sediment of this particular area, right? But if you are interested in understanding the subsurface microbiome, or let us say a different section of the subsurface, then you have to use corers. So corers will look something like this. Okay, and here when you are putting the corers in the sediment, it will collect the length of whatever the length of this corer is. It will collect the sediment from that for all the length, and then you can make a fraction that if you want this 5 centimeter, or if you want in 10 centimeter, likewise you can take that particular environment and then do your microbiological work. Okay, if it is happening in a shallow area, then you can do like this. But if you are going in the open sea for collecting the deep sea sediments, then you have to go with this kind of big toe tie, and then once it goes in the surface, it ejects the corers, and then the corer will have bacteria or let's say microbes for the different subsurface in the sediments. Okay. So the first one is for the water column, and the first one is mostly used. This plankton net is mostly used for phyto and the zooplankton, and the microbes, I mean bacteria or archaea associated with the phyto and the zooplankton. The second one is for the sediment samples, so this grab and corers. Now the third one is a Niskin sampler. So it's a kind of plastic cylinder that are open at both the ends, right? What you do is that you tow it down in the water for collecting the water column, different water column. And let us suppose that you want a 10-meter deep sample, water sample. So you put this Niskin sampler inside. Once you reach 10 meter, which will be marked in the thread also, right? Once you reach there, you stop it, and then you put a weight. So this weight can be either sensory, I mean, included with the electrical signal, or you can also put directly, manual weight. Once you put the weight, it will close the lid from both the sides. Okay, and now you have 10-meter deep water sample in this Niskin sample. Okay. So for any microbiological sampling, what we need to remember is that we have to use a sterile plastic bag, sterile bottles, and taking care of avoiding any contaminations. So since we know that microbes are present everywhere, if you are not doing the sampling properly, then what is possibility that instead of taking a water column microbes, you may grow or you may work with surface water, or you may work with contaminants microbes, right? So that is the first important step in the sample collection. And second is that the ocean water is highly heterogeneous, right? It's a quite mixed system. It's not a homogeneous system, right? So you have to take a proper replication, frequency, and location of the sample. For any marine microbiology, there could be two possibilities. One, if you are working in a coastal water, sediments, or the intertidal area, which is a straightforward because you take those grab, cores, or skin in a small boat, or let us suppose go with a scuba diving and then collect the sediment or water column you want. Or if you're going in open ocean, right, or you want in a deep sea sediment, you have to go in a research vessel, right? So the research vessel should be equipped with the gears and the onboard laboratory facilities. There are plenty, so we don't have to worry if we are going in the open sea to collect the water column or the deep water, right? When you are going in the open ocean or in the research vessel, you have something called as circular rosette system, okay? Also called as rosette, okay? So this rosette looks something like this circular system where you have Niskin sampler. So in the past slide, if you remember that I showed you, there is a Niskin which is having open mouth, I mean open both end, right? And then once it goes to a particular water level, you want to collect, you can close it, right? But it is difficult when you are going in the open sea. So it is fixed in a rosette system where close to 24 Niskin samplers can go all together at one time. And this system is also attached with a sensor for conductivity, for temperature, and for the depth. And in some certain cases, you can also have the parameters like turbidity, dissolved oxygen, fluorescence, and photosynthetically active radiations, and whatever other sensors you would like to add in this, right? So that is a those factors are very much important when you are working with microbes, because if you would like to know what would have influenced your microbial community in that area, then these abiotic factors are very much important. So in the rosette circular system, you have all the sensors, and then you have 24 Niskin samplers, The benefit is at the same time you can collect a different depths, because you can close, let's say, two of this Niskin at 5 meter, then further you can go down and two of in 10 meter. Likewise, you can collect different uh, depth of water column in one sampling itself. Okay. Also, when you go in a research vessel, then for the planktons, there is a net, big net, very big net, which drags in the bottom of, I mean, in the back side of the research vessel, and it drags as the research vessel moves, and then it helps in the collection of the planktons. Okay, so this is the system that how you should do the sampling for the marine microbial studies. Now, what is the second step? That if you get the water from the Niskin sampler, or forever, like let's say that you just want to see the microbial community in the Marina Beach. So you went there, you collected the sample, and either in the field itself, or if you are in the research vessel, in the vessel itself, or after coming back to the lab, right? You have to filter it. So for filtration, we have two ways. Either you use the vacuum pump based filtration unit, right? So you have a filter paper here, and a locking system with the with a place to uh, for the water inlet, and then water will be filled here uh, using the vacuum, right? And then the microbial cell will come on the filter, right? And then there is another way to do the filtration, that is a peristaltic pump. So in this there is a no pressure on the water, because peristaltic pump will pump the water in this direction, and then in here you can put the filter, Sterivex filter. So whatever the microbes are there in the water column will go and filter here, right? So the advantage here is that there is no pressure on the cell, so cell damage can be less, but the speed is slow. In here, you have a vacuum pressure, so there are chances that some of the, if you put too much of vacuum, then there is a chance of the cell disruption, but the process will be faster, right? So these two common methods for the water filtration for microbes in the in the marine microbiology. Also, if you look into the open ocean, then there is something called as remotely operated vehicles, or called as ROVs, right? It is a kind of robot, as you can see in Woods Hole Institute, one of the ROVs, that this goes down. It is having robotic arm and all the sensors, which can be operated from the boat itself, from the research vessel. It can collect the water or the sediment sample in the deep sea, right? Another method which was invented in 1931 by Elster Hardy is a continuous plankton recorder. So continuous plankton recorder, what it does is that you can tie it to the bottom of the ship, okay, and then it can continuously uh, collect the samples, which after the boat stops, or the ship comes back to the shore, you can remove and then you can analyze. Okay, so this is continuous plankton recorder, right? So these are some of the methods that how you collect the water sample and the sediment sample. So we have plankton nets of different mesh size. We have grab, we have coarse, we have Niskin water sampler. This Niskin water sampler is linked with all the other probes, mostly CTD, that is conductivity, temperature, and depth, and it is called a circular rosette system. If you are in the open sea, then the plankton net can be linked with the backside of the research vessel, which tow it while running, right? And then when you get the water sample, you have to do the filtration. That can be done either with the pump-based method or with the peristaltic pump, right? And there is another method for remotely operated vehicles, which do have the arms and which do have the cables, which can be operated from the surface. Then the last one is continuous plankton recording. Now, utilizing any of this water or the sediment sampler, you can perform or you can study the microbes present in the marine environment. In the sampling method itself, it is important to tell you that there could be two particular, actually there could be three particular type of studies. One is that one-time study, that you went at one location, you studied, and you collected the organism. You went, you focused on the biology of that organism, and you never went sampling again, right? That is first. The second one, in order to understand better the system of this microbial community, their functions in the nature, there can be a temporal studies. So temporal study is a study where one location you study for a continuous long time, right? So you collected sample, let's say today, then again you collected tomorrow from the same location, and likewise you do it for years, right? So that is a temporal study. For doing the temporal study across the world, we have several long-term ecological research network or long-term ecological monitoring sites. Okay, in this long-term ecological monitoring sites, people have been investigating for last 30 to 40 to 50 years. What are the abiotic factors there? So including all salinity, pH conditions, what all the nutrients in that particular area, uh, uh, nitrogen, phosphate, silicate, and so on, and also a number of biological parameters like phytoplankton biomass diversity, benthic invertebrate diversity, fish abundance, zooplankton, macroalgal canopy, or microbial biomasses, right? Turtle aggregations, seagrass canopy, hard corals, and mangrove covers, right? You can see that across the world we have this many number, or you can say that these many locations or the habitat where the long-term ecological monitoring has been happening. Estuary, coastal lagoon, sandy beach, coastal wetland, and so and so, right? In this particular. If you want to go and look more that what all the studies have been happening in the LTER, that is long-term ecological monitoring sites, one link I have given here for Estación Zoologica, where they have been maintaining a site called as Mariquera near to Naples in the Mediterranean Sea, and performing for last 40 years studies in order to understand the distribution, diversity, and the function of the organisms at that particular location. Okay. Likewise, you can see a lot of there. Unfortunately, in this particular paper, or as far as I know, there are not many long-term ecological sites are there in the Indian waters, though there are sporadic works happening in NIO and several other central and state universities where they are studying the sites. For example, even ISA Kolkata has been studying one site in Sagar Island for close to 10 years. So likewise there are, but then there is no international research network for the long-term studies. Okay. Uh, so similar to the temporal study, there are also attempt made in order to understand at one particular time, so kind of a special study in the ocean at one particular time, that is mostly on the June 21st in 2014 and 2015, that is a summer solstice, right? So on the day of summer solstice on 21st June, at a particular time across the world, the sample have been taken from different seas, okay, for the next generation sequencing, in biodiversity analysis using the next generation sequencing technology, in order to know who is out there, what are the functions they are doing, and in order to get more and more information on the marine microbes. Again, I don't think that there was any sampling in the India. Okay. So as I said that there could be three kind of studies. One is one-time study. Second is temporal study, that you keep on going at one site in order to understand better the marine microbes. There could be the third one. So it should be special. It's wrong. So I will correct this. So special study. So in the special study, what is happening? A number of expeditions are going, as how it went for Darwin, right? Similarly, for the especially focusing on the marine microbes, there are global ocean sampling expedition. Again, this one is promoted by the Craig Venter, right? And then you can see that from 2003 to 2008, and then 2009 2010 in Europe. So this is the path that what it took, the research vessel took in order to collect the microbial data from the different part of the ocean. And the one example I gave already about the Tara Ocean, which close to 10 years collected the sample in the different part of the oceans. Okay. Now coming back to what we have in our country, as I said that CSIR NIO is doing some of the microbial studies, holding some of the research vessels, which goes in order to study the marine microbes of the tropical Indian Ocean up to the equator, and also a very frequent research vessel going in the east and the west coast of India, understanding the microbial diversity and across the surface and the in the deep water as well. Okay, and also they have been sampling frequently oxygen minimum zone in the northeastern Arabian Sea. Okay, uh, most of the research vessel in India is coming under the Ministry of Earth Science, because most of the organization, that is NCCR, that Coastal Conservation Institute, that is CMLRE for the Marine Living Resources, NCPOR, that is the Polar Research Institute, and then INCOIS handling the coastal oceanographic data, and then National Institute of Ocean Technology (NIOT). All these institute comes under one umbrella, that is Ministry of Earth Science, and they do have a separate division for microbial oceanography, where they have been studying very frequently the roles of the marine microbes in the marine food web, nutrient cycling, formation of oxygen minimum zone, planktons, picoplankton, archaea, bacteria, actinomycetes, fungi, and all occupying all the realms in the oceans. Okay, from the sediment to the pelagic conditions. And also, as I said, that MOE is having various research vessels. For example, Sagar Kanya, Sagar Sampada, Sagar Nidhi, Sagar Manjusha, Sagar Purvi, and Sagar Paschim. Right? One such image you can see here. I think this is Sagar Kanya. Okay, and you can see that here mostly the nets will be there, plankton nets will be there, and apart from the access in the ocean for the marine microbiology, we also have a dedicated institute for the polar and ocean research, uh, which conduct a regular expedition in Antarctic, Arctic, Himalaya, Southern Ocean, right, and then international cooperation for ocean programs. Okay, uh, they are also frequently visiting to Antarctic and the Arctic, so we do have the possibility to conduct the polar microbiology using the Bharti and the Maitri at Antarctic and Himadri station in the Arctic. Okay. Apart from these two locations, I should tell that in NIOT in Chennai, we do have a facility. I don't know whether it is functioning at present or not, but last time when we visited, it was working as fermenter to maintain the uh, high pressure for working on the deep sea microbiological experiment. I thought it is a very interesting information to give you guys in order to know that we do have the facilities to perform deep sea microbiology, polar microbiology in the extreme conditions, and also we do have the possibility to collect the oceans and the sediments in the through these research vessels in India, right? So we have covered the sampling strategies or sampling methods for the marine microbiology.